community?
Yes, sir. We all are good, sir.
Great to hear. Great to hear.
Yeah. Yes, sir. Yes, sir. Can I start the welcome, sir? Can I start the welcome?
Yes, absolutely. Absolutely. Please go ahead.
Yeah. Thank you. அனைவருக்கும் வணக்கம். It gives me a great pleasure to extend you all a very warm welcome on the behalf of the management of Kalasalingam Group of Institution and with the support of our founder chairman Kalvi Vallal Deivathirti Kalasalingam Ayya and the support of our chairman Ilaya Vallal Dr. S. Sridharan sir for the international virtual learning series titled on lipid based formulation. The expert talk on lipid based formulation in association with the IPA South Tamil Nadu local branch, head office at Arulmigu Kalasalingam College of Pharmacy. Today we are proud to have an eminent scientist, distinguished personality, well-known administrator, and honorable resource person, chief guest, and resource person. Can you hear, sir?
Yes, I can hear you. Thank you.
Thank you. Chief guest and honorable resource person, Dr. Indranil Nandi, Senior Vice President of President and Head R&D Research and Development, Jubliant Pharma Limited, USA, to grace this occasion. It is a great honor to have with us for this occasion. Thank you, sir, for kindly being the valuable part of this session. Your prestigious presence is a great honor for us and great motivation and inspiration for our students, the entire students community. Sir, we are fully appreciate you for finding the time to be with us despite of your busy schedule, sir. Most welcome, sir. Most welcome you, sir.
Thank you, Dr. Venkateshan.
Let me acquaint you with the sterling quality of this eminent personality who is a well-known researcher. Thank you, sir, for accepting our invitation and spending your precious time with us. Sir, most welcome you, sir. Most welcome you, sir.
Thank you, Doctor. Thank you very much. Thank you very much for your kind word.
Thank you, sir.
So, shall I?
Yes, sir.
Okay, thank you. So, first of all, thank you, Dr. Venkateshan, for such a kind words, and thank you the college for inviting me, and congratulate the college administration for arranging such a talk for the students and the fellow pharmaceutical scientists in amid of pandemic in the country. And it is a great initiative, and I wanted to congratulate the college, and I wanted to congratulate all of you who are joining the call on a Sunday evening. So I think that itself shows a lot about the commitment all of you have in pharmaceutical sciences. So I think that itself speaks for itself. So I wanted to start saying congratulations on that. So with that, what I would like to do is spend some time on a on a presentation which is basically on formulation, or what a lot of students known as a pharmaceutics side of this business, and talk about some of the things what we do on a new chemical entity development. So we all talk about the, in India we do a lot of generic drug development where the molecule properties are known, and kind of you get the data from this literature, and you kind of from the patent or the literature you learn certain thing and you try to do. What I'm going to do talk today, when we are at least at the first half of the presentation, when we discover a drug and we kind of have zero idea how the drug works, how we decide what to do to deliver the drug to human body, and to make sure that first in man studies are reasonable enough to go to the next phase. So that will be the most part of the discussion. So let me share my screen. So just a quick clarification. Dr. Venkatesan, can people see the screen, the PowerPoint presentation screen?
Yes, sir. Yes, sir. Visible,
okay,
sir.
Fantastic. So, as I was saying, we will talk about a formulation which is based on the lipid, and we will try, based on the time we have, we will try to spend certain time on first in man, and then to see in other dosage form, which is not really a traditional oral route of administration. We'll go into some of the transdermal and nasal administration and show some real-time data that actually work on the that use the lipid formulation. So before I start, I wanted to put a pure disclaimer. This represent personal view of the presenter, and it does not contain or reflect any opinion of Jubilant Sciences or its affiliates. So I wanted to start with that disclaimer, and then I can talk to you about what we are going to do. As I said, I'm going to do a little bit of introduction, why we are talking about the lipid-based formulation. Then I will talk about how we go into the first-in-man formulation. And then we will talk about a little bit of microemulsion applications. Then we will go one or two case studies, transdermal delivery and an intranasal delivery. And then we'll finish up the conversation with the future challenges. And if we have time, we can talk about a little bit of case studies for the group. So what I would, and I have no idea, this is a fairly large presentation, so I'll try to keep it in a reasonable time. So it's around 5:30 on us, 5:40 on the Sunday evening. So I know that it's a day off, so I will try to finish it off a reasonable time and keep some time for a little bit of discussion. So hope that's the game plan, and let's see how our time permits. So what I wanted to start, why we are suddenly talking about lipid formulation, why it is very interesting from a new drug side. So the challenges that we see in the new drug side, we see that majority of the drugs we see are poorly water soluble. So when you give a poorly water soluble drug in our body, particularly if you do oral route, what you are going to see, poor and highly variable bioavailability. And we also can see a little bit of food effect and a first effect, and you will see suboptimal dosing and performance that required to get the therapeutic efficacy. So for that reason alone, we needed a, a alternative dosage form, which is which which is which is a traditional tablet or other stuff. So give me a second. So before we go there, and when we look at the drug, we usually classify the drug in biopharmaceutical classification of drugs, which is BCS classification. We traditionally on a new drug side, we try to be put all the drugs into BCS class 1, 2, 3, and 4. And each of the classification has its own meaning. Those who are aware of the BCS classification, they target the product based on the behavior in the oral gut. So the first classification is called BCS class 1, which is aqueous, highly soluble and highly permeable drug. That means it not only gets solubilized in the gut, it also easily get permeated to the gut membrane. And then the other part of the BCS classification is class 4, which is low in solubility. That means it doesn't get dissolved into the gut fluid, and it doesn't get permeated at all in the system. But the interesting part, what you will know, the class 2 and the class 4, these are the two class where 40% of the new chemical that we discover in the world are actually falls into. And this is where you see the low solubility of the drug in the gut fluid really plays a vital role in how much drugs are actually getting absorbed into our body. So going forward, so what we landed up doing that, as I was talking, that when we are having the low solubility, it leads to the poor bioavailability. And what you are seeing, there are three sets of problem that primarily affect the drug absorption in the gut. So what are the three types of challenge we see? One is physiochemical problem, which means the drug is not soluble, as I told in the previous slide, and poor diffusion to the intestinal membrane, that is with low permeability. So that is the one part of the problem. Then the second part is significant first part hepatic metabolism. This is a challenge that you cannot overcome with any sort of formulation if you wanted to do it on the oral route. If your product has a first pass hepatic metabolism, you need to kind of bypass the oral delivery route, and you have to look for an alternative route. For example, you have to look at for injections. You may have to give it for transdermal, or you have to give it for pulmonary or nasal route. So for the second part of the problem, as a formulators on a new drug side, we tend to go to alternative dosage form. And the biochemical processes in the intestine, that is metabolic enzymes and efflux transporters, these are all very complex system and definitely beyond the scope of this talk. If those are affecting, usually the drug available in the market tends to go for a route which is non-solid, unless they are enhancing the uptake mechanism. So today, what we are trying to talk about, the how to solve the physiochemical problem. That means what we can do to improve the solubility, and then later part we will show how we go to a different formulations other than oral formulation to bypass the hepatic metabolism. So on a new drug side, these are the typically two of the biggest challenge as a formulators people usually face. So just to summarize those who are fairly new on the pharmaceutical world, that how the absorption process works. And this is for the students, third year, fourth year students, kind of very simple schematic diagram that I am showing, that what are the drugs goes through without going into the much complication. So drug goes to the dissolution, then it goes to the membrane permeation, and then it goes to the systemic absorption. And each stage, if you see these sections, there is a way the drug get destroyed. So one case is drug is eliminated in the feces, whatever not dissolved. During the, let's say it get dissolved, go through the solution, it still go through certain degradation, like chemical degradation. Maybe it's not acid sensitive, it may be acid sensitive, or it might be basic sensitive. So there's a chemical degradation. Then it can go to the luminal metabolism. That means it get metabolized into the system. And again, it can still go through the feces even after dissolution if it is not getting through the membrane. And then when it goes to the systematic absorption, then it still go through two sets of degradation, or you can say elimination, intestinal metabolism and hepatic elimination. So you can see there are many things can happen when the drugs go through the whole process of going, when you take a tablet and when it actually works in your body. And you can take any molecule, as simple as taking paracetamol, to as complex as today's drug, say remdesivir. What will happen to it when it goes to the body? And so next part of the topic, what we are going to do, we are going to talk about the physical support, right? So in the previous slide, I showed about that we will be talking about the poor solubility. So primarily focus on the solubility and the dissolution. So when we are talking about the dissolution, so there are three types of barriers in the drug dissolution. So the first barrier is the crystal energy and intramolecular attractive force of the molecule. So the molecule itself is such a complex molecule that it will not allow you to go into the dissolution. Then the second thing is that molecular composition of the drug substance. Maybe the drug is highly hydrophobic. That means it doesn't want to go into the solution. Then you will have a serious problem with that, you know. And then the last piece is maybe the surface area. The surface area of the molecule when you are putting into the tablet may not be sufficient to expose to the dissolution media. And that itself has a problem in the barriers. So all these three are real-time barriers that you are going to see. So what we do, and this is a very complex chart, and I will try to explain to you what one can do. So let's say we find a molecules and we wanted to formulate, and we kind of go through our decision tree. So we go through each type of decision tree. Let's say we go saying that non-ionizable group and ionizable group. We first look into the drug and see whether it has an ion or ionizable group or not. Let's say if I have a ionizable group, and definitely immediately we go to different type of salt. Let's say we will get succinate salt, oxalate salt, different type of salt, and then we do the develop the product in the salt form, which is actually pretty easy in the molecule development point of view. The challenges become that when there is a product we develop which does not have any ionizable group. Now, and this is where the majority of the molecule comes, then we kind of go through the different decision tree. So the first decision tree, is it a high dose drug or is it a low dose drug we are targeting? Some are very potent compound, let's say anti-cancer drugs. We probably will be able to do certain thing, but some may not be. So people kind of goes to the high dose versus low dose, and depending upon if it is high dose drug, one of the first thing we try to do, reduce particle size to see the surface area, and then if the surface area is helping, then we will use the approach called using particle size. But then you might actually find in the rabbit or other animal studies, it is not improving. Then we go into the three types of alternative formulation: lipid, complexation, and solvent. So there are multiple approach you can do, and we are only talking about the lipid-based formulation today. And the same way on a low dose side, we look into the lipophilicity and the activity of the how hydrophilic or hydrophobic the drug is. And if the drug is really low lipophilic and non-ionizable and low dose, our traditional approach on those products is not to take that molecule at all, because we know that nothing will work to improve the drug dissolution. So in that case, let's move on to the next molecule, and we'll focus on the product that has a little bit better lipophilicity and all this thing. So this is a decision tree. We different company fellows. It is one of the decision tree. There could be hundreds of different decision tree we can follow. Okay, so with that, what we do for next few slides, I will try to address each of the methodology that we do on the new drug side to address these issues, each of this approach on the formulation. So first one, obviously the very simple one, which is a particle size reduction. So that means you mill the product. You get a particle size of, let's say, 250 micron, and you basically take it to the 10 microns and it works. Why it works? Because you basically increasing the surface areas. By increasing the surface area, you are just improving the dissolution rate for the product. And it usually have a positive effect on the hydrophobic pharmaphores if you do surface area. The challenges are once you started doing the micronization, depending upon how small do you go, that means how small particle size you go, you might landed up being in a situation where the molecules started to aggregate because of the potential surface charge, and that's a real problem. So if you look at the many nanotechnologies that we do about the micro nanotechnologies, where the particle size go below 10 micron, 5 micron, 2 micron, one of the challenges you are going to see that aggregation over the time. And that's a real challenge. That's a real physical chemistry, and that takes a lot of time on the scientists to figure this thing. And you will see a lot of publication, a lot of work, a lot of patent on that. But nevertheless, this technology, particle size reduction, is a very common process on a new drug side. In fact, I know companies where they have a standard protocol. Get a molecule, a new drug synthesized by Metchem. First thing you do, it is mill the product. Just don't bother about anything else. So there's companies who have done that. So then required the next part. Remember, we talked about salt formation with a molecule which has the ionizable group, and this is also a kind of a strategy that a lot of people follow. So the first thing, of course, as I said, it requires a ionizable functional group, and we need to prepare a salt that has an acceptable physical property. And this is a very important piece, right? So you are preparing the salt to make sure that it is not, it can withstand the oral dosage form. For example, it has a sufficient melting point to withstand milling or formulation processing. Let's say you develop a salt, which is a very good salt, but the salt kind of melt in 40 degree or 42 degrees Celsius. What you will see in a phase 2, phase 3, when they are making the tablet and they are putting the drug into the wet granulations and they are trying to mill the product, there will be huge problem in terms of the formulation of that, at that time, processability of the product. And the most important thing is that identify the stable crystalline form. Whenever you create a salt, you have to figure it out, what is the stable crystal that can stay like the salt, and it will not create a different isomers. It will not create other problems. And it can be done in a high yield. That means when you make a salt, it should not be when you start an experiment, you took a 1 gram of base and you created a salt, and salt is only 5 milligram. That's not yield, proper yield. That means you are not able to create a good reactions. And that's not the right thing that eventually goes into the market. So always when scientists look into the salt formation, their target is you get a reasonable yield, which I would say 75 to 85% yield against the base molecule to get a product that is acceptable for, and it has to be stable. It cannot be fluctuate. So these are the parameters we as a scientist, when we go into the new drug product development, we kind of manages this, And the other challenges with the salt formation is that it's pretty much you start the clock from the beginning, because the moment you start the salt formulation you created a stable salt, you have to redo some of the pre-formulation work, safety studies, and the efficacy studies again, so on and so forth. Okay. And the next approach would be 1 second, quick. Dr. Venkateshan,
Sir, please,
one quick question. Is there a question answer session? People wanted to ask question in the middle, or how we are going to do it?
After session.
Okay. So no question in the middle. Okay. Okay, perfect. Let me continue then. Okay. So the last part, next approach that we kind of do is a complexation. If you remember on the graphs that I showed, there was a process of particle size, salt selection, complexations, co-solvent, and the last was the lipid formulation. So there are total five way you can address the physicochemical issues that we see in a new molecule. So the third approach is a complexation, where we are seeing what is complexations. We are talking about a most common complexation. What people try to do is a cyclodextrin complexations. So the advantage of such complexation is a very reversible system, and it's a very predictable and reproducible. And one of the thing is that it required a ligand in the formulation, and it's a very specific interaction. So think about cyclodextrin. Cyclodextrin, when it goes into the solution, it's almost create a cavity, and the molecule kind of goes inside that cavity to go into the solution. And it's not true for everybody, right? So based on the molecule size and cavity size, it may not work at all. And we can do all those research at the very beginning itself. And we can say, you know what, complexation will not work just by the size of the molecule that has been reported by the MedChem team. And then, of course, the other piece is that it also create it has an impact on its drug stability. Some products become much stable in the complexation, some do not. So we have to study that thing very carefully. And the good news about the complexation is that complexation is a very universal form. So it's just not used for, it is highly used in a injectable form to solubilize the product, and there are cyclodextrin, which are very known to use in the injectable form. It also used in the oral form. It also used in other doses form also. So that's the beauty of the cyclodextrin, is that it's a purely reversible system and used in a multiple form. And it is unlike the salt form, you really don't have to start the efficacy studies or safety studies from the beginning, knowing as long as you know that how much cyclodextrin you are using, because it is reversible and it is used in the as an excipient. So I think the last before we go to the lipid system, one more system that we mentioned is a co-solvent. And the co-solvent system, probably the very common system, you know that the drug is very soluble, very insoluble in water, but it has a good solubility in ethanol. So what people might try, they try to make a compositions of water and ethanol and see if that is workable. Same thing, they try composition of PEG, propylene glycol, and water or something else. And these may work as long as they are oral liquid form, but it may not work when you are talking about oral tablet form. So the co-solvent application kind of goes limited depending upon whether you wanted to make it pure liquid or not. So that's why there's a certain limitation of application of co-solvent in the product. But it is highly used in a phase 1, phase 2 study because of the simple reason it is easy to manufacture. So the one other application that we are going to talk today is lipid-based formulation. So the last part is using of a lipid. So why? And then there are different type of lipid systems are used. So they use lipid solution and self-emulsifying drug delivery system, then self-microemulsion, suspension, semi-solid, regular emulsion, solid dispersion. These are all the system that used into the into new drug site using lipid to deliver the drug into the body via oral route and able to get the efficacies as good as possible. So what is the advantage of this? Disperse, solubilize, and maintain solubility of the drug in GI fluid and influence the gut wall permeability. And that's a very important. Since the lipid membrane, lipid is a part of the membrane structure itself, it has its own unique way to interact with the membrane wall and hence kind of help drug to be permeate better. So let's talk about a little bit of each of the system, what I'm defining in the last slide. So the first one is a lipid solution. This is of the very, very basic thing people do. The drug dissolves in a lipid vehicle and it forms a crude emulsion on exposure, aqueous media upon mild agitation. So this is the crudest form, and you just create that lipid solution and give it to as long as it works, it might be a good system. The self-emulsifying drug delivery system is an isotropic mixture of drug, lipophilic mixture of surfactant, which form emulsion or microemulsion with the aqueous fluid on mild agitation. So this is a little bit more complex system, and it has its own advantage. It's create a emulsion It comes as a oily stuff when it goes to the water, when it goes to the gut fluid, and it creates the emulsion, and it helps into the system, goes into the body. So, so usually what we are looking at the different component that we are using in the lipid-based formulation. Of course, there's a drug, and then oil or lipid. Hence, it's a lipid formulation. And then the third part that which usually plays a very, very important piece is an emulsifier and co-emulsifier. So as those people who have done emulsion preparation in the laboratories, I guess it is a mandatory in third year pharmacy, if I remember correctly, third year or fourth year, where you make benzalkonium emulsion or some other emulsion. So if you remember, one of the thing it works is having the right emulsifier. So that helps you to create that emulsion. So now think of taking a formulation which is pre-mixed with the oil and the emulsifier, but the way it is done that as long as it goes into the gut and mix there with the water, they immediately form an emulsion. And that emulsion will entrap the drug inside, and it will slowly take it to the gut wall and able to help it to help the drug to permeate through the system. So as I was telling that what is happening with the lipid formulation, first thing is the oral bioavailability got improved. It improved the content uniformity of the low dose. That means it get uniformly dissolved into the oily system. Therefore, you don't see any issues in the dosing. It also affects on the absorption barriers, as I was talking, that is actually interact with the gut wall because they are also made of lipid, and that helps to improve the absorption barrier. So what is the challenge? So obviously the question becomes, can we use any oil? So one of the art of this technology is to identify what lipid-based system we should use. And then there's a very minimum in vitro, in vivo database, which kind of make it hard for a lot of people to figure it out the right lipid system. Having said that, there are most companies has their proprietary database that they use to do their own work. And then scaling up, this technologies, as you move from one place to other, they might be very good to work in the laboratory scale in a small beakers, but as you go up, it might be absolutely impossible to scaling up unless you have a full control on that. And then source of the lipid, and one of the things that we do when we are talking about the source of the lipid or excipient selections, it must be approved for human use. This is a very important piece. The molecule, the excipient, the lipid must be having a GRAS status, or there's a market precedence in regulated market where the product, the molecule has been used before. And this is really depends on the company policies. Consistency in quality, uniform performance for the given application. So one of the challenges in the lipid, whenever you go to a formulation world, you will see one lot is very good, the second lot is very bad. And then comes to the control specification, that is it a good, the molecule is properly, is the excipient company knows what they're doing, or they're just making just for sake of making, And then you have to be very careful and have a very tighter excipient specification and impurities. For example, lipids are known to have a peroxide impurities, and that peroxide impurities can create a haywire in the drug stability itself. So you need to have that. You need to have that foresight that this is what's going to happen. It might create an oxidation problem for my drug, and you kind of plan these activities from the day one. So as we are going further and further, so there are a few other things we also pay attention. So excipient drug stability over time, temperature dependent solubilities, and aging polymeric issues of the lipid. So this is a big problem. And so let's say companies get a big drum of lipid. Usually they come in a drum, and then they started using a smaller portion out of it. Over the time, the lipid actually loses its potential and the behavior totally changes. And that's called aging issues with the lipid. And this is a huge commercial problem. And so most company pays very close attention to that and manage accordingly. And when, as a scientist who developed the product in the lab, they have to kind of tell to the manufacturing team that, guys, please careful about this. Don't use a lipid beyond this much and this much. And it has its own implications. And one of the things is that as a scientist, we should be always aware is the awareness of saturation. Supersaturation status of the lipid has a crashing out effect. So this is a very important. So, if it is a highly lipophilic drug and you are taking a lipid, the drugs are kind of dissolved into the system. Now the scientists get excited. They try to use a minimum amount of oil and maximum amount of drug loading. And it might lead to the supersaturation. So one of the first thing the team should be doing, kind of figuring out what is the optimal level. Because if you don't do that, those are all formulation work people have to do to make sure that you created a great system, but you actually created a supersaturated lipid system. So it's great at the beginning. And this is for people who are going to do PhD thesis on a lipid system. It's a very common mistake. We call it rookie mistake for the PhD student. You kind of create a system. It looks good. You started doing all the experiment, and suddenly you started seeing everything started failing. The stability started failing. The clear solution of the lipid started to become hazy. And that's because the drug is crashing out. So you have to do the initial study very carefully, and it saves a lot of time later on. And as I was talking about, from a oil point of view, you can look at the different corn oil, monotriglyceride, medium chain fatty acid. Surfactant, we can usually look at 2,80, chemophore, PEG esters of the fatty acid, and co-solvent, some of the things we can use, transcutol, even ethanol. Certain cases, certain amount of ethanol is useful, and we can try those. So with that, we are 45 minutes into the presentation. So what I would do, I will try to wrap up in next 10 minutes, and we'll close with the first case studies. Dr. Venkateshan, hope that works for you.
Yes, sir.
Okay. So I will try to wrap it up in next 5 minutes, and then we'll go from there. How about that?
Yes.
So this is actually a trial that shows a very promising efficacy in animal studies. That means preclinical trial, and the efficacy is high. The people think that this is a real breakthrough molecule, and we wanted to go to the first in man studies, and we have to create a formulation that is suitable in first in man. And for that, you have certain information. We usually get certain information. So what we got that what information we have in our head is that it's insoluble in water. First question. That means drug is probably highly lipophilic. So if you remember the chart, it kind of goes into the lipophilic, and it's a low dose. Remember the two things that people are thinking. And it's also unstable in presence of water and metal ion. Very important information. It also do hydrolytic cleavage. It is stable at neutral or acidic pH. And carboxylic group in citrate buffer catalyzes degradation. Why this information? These are looks very unrelated information, but it is all connected with the decision tree people will be taking. So what is insoluble in water means? Insoluble in water means that it is most likely it's a fairly lipophilic product. It is unstable in presence of metal or water and metal ions. What does it mean? That we cannot use a co-solvent system, because if you wanted to do a co-solvent system, you have to use some water. And if you use some water, it will immediately degrade. Therefore, you cannot use a co-solvent system. And it also degrade in presence of metal ion. This is an important piece, because think about it, right? If you are doing metal ion, where do you think that you will see the metal ion? If you're trying to milling, you have to use a metallic machine to do the milling process. So there's a likelihood that it will create impurity during the milling process. So the kind of particle size goes out of the process as well. It is stable in neutral and acidic pH. What does it mean? That means as long as you somehow deliver the drug and it solubilizes into the acidic, which is the gut, or neutral pH, that means it is intestine, small intestine. If you are up to small intestine, it will be stomach and the small intestine, it will be stable. That is a very important. See how we are connecting the data from the drug to our body. And this is the most important part of a drug discovery formulation for new drug site, connecting the physical data with the body's condition in the body. It's a simple data, but it has its own impact when you are thinking how the drug will behave in the body. Light sensitive, that's what it is, right? So we have to be careful on the manufacturing point of view. With that, what we started to do, went back to this whole section. Remember that we told you that there's a decision tree. So we went to the ionization and non-ionization. So there's no ionizable ground because it is getting through the hydrolytic cleavage and the neutral pH. So we basically got rid of this section of this. As you see my laser pointer, this section we just forget about it, right? Then we went to no ionizable drugs, and it is not a high dose. So all these sections kind of goes away. So now it goes to the low dose. And once it low dose, we know it is not soluble. It's a highly hydrophobic, highly lipophilic. So in that case, these sections, again, we cross out. Then we started looking at the alternative approach. What are the alternative approach? We are basically started to look at this complexation based on the hydrolytic cleavage and other thing. Complexation doesn't work, but it is more of a other factors we have seen. Now there are two aspects: lipid-based formulation or co-solvent. As I pointed out, if you take it a co-solvent, this drug is highly unstable in the water system. Therefore, this is also crossed out. So basically at the end, we landed up making a decision, we have to approach this formulation lipid system to make this product actually work in an oral route and give it to the body. Okay, so with that, I will stop my presentation at this moment. And because if I go to the next round of the discussion, I have to spend another 45 minutes. So I will stop here and I will wait for some questions if there's any question on the presentation so far.
Questions? Any questions? Professor Anburaj?
Yes, sir.
Is there any question?
So, sir, there is any artificial intelligence tool to predict the type of dosage form, sir, from the pre-formulation characteristics?
Look, we are all working on machine learning. So the data, there is tons of data there. And there are work done on artificial intelligence, and companies are using AI to predict the molecule. In fact, Google has its own group right now who are using the AI to improve the drug discovery. So yes, there is a huge potential of that. It really depends on the sample size, what you have in your database, and larger the database, your machine learning will be improving significantly.
Thank you, sir.
Okay.
Sir, you have told that emulsifying systems having a lot of stability issues.
Yes.
So how it will be successful to formulate a self-emulsifying system, sir?
So the one of the way you look at it, certain molecule cannot be done oral route. It's the reality of the thing, right? So you have to look at the molecule at the pre-formulation stage. If the lipid, are they interactive with lipid so much, is that a lipid purity has an issue? See, as long as the lipid itself is not an issue, right? It is the other stuff inside the lipid may have been creating the problem, as I told about the peroxide impurities, sort of thing. So if you are able to manage that, you will be able to successful. So if you know that product is highly sensitive to oxidation, probably you might see some real issues in the system itself. Let's say if you are not doing the phase diagram properly, you might see a real problem in that case. Maybe I can show you some phase diagram. Let me see if I can show. So one of the thing you can definitely able to do better job if you do a real-time detailed phase diagram like this, and you actually know where the emulsion will be stabilized. You can do complex phase diagram where you will find a emulsion zone, where you will find a microemulsion zones, and each of those zone might give you a very specific, stability, and that's how you probably can able to manage this piece.
Thank you, sir.
Yeah.
Sir.
Yes, please.
So actually, you dealt with lot of pre-formulation data, sir. in Indian scenario, normally we are going to deal with mostly with generic formulations. So we are going to get a free formulation data from literature or some other data sources. In NCE, New Chemical Entity, whether it's a role of formulation scientists to predict the pre-formulation studies or otherwise that molecular development people they will give it us for us.
No, so you get a very basic data from the medicinal chemist, but you have to do most of your work by yourself. I I just saw something on the chat. Let me ask. So Shomia wrote a note saying that they're doing two tertiary diagram to microemulsion zone. It is taking too much time. Is there a software available to find optimum concentration drug delivery ratio? So it's a great question, Somia. So as long as the drug is unique, you might get a crude idea. There might be some software available, but it is a very it takes time. It takes time. I cannot. I agree with you. It's a lot of experiment, but you can do a smarter experiment. I don't know what is the total number of experiment you are doing. You actually can create a decent tertiary diagram with let's say 15 to 20 data point. I hope that is not too much data point for you. So and you need to still have to input certain amount of data to software, even create that diagram. Anything less than 10, probably it will not come with the theoretical data, because of the simple reason drug solubility is always an issue. So your each drug solubility into the each system is a very different, right? So you still have to do the drug solubility study to predict it. So I think 10, 15 data, 15 data point would be reasonable. Hope, Shomia, I was able to answer that question. One question, someone called Guest said, modern technology, what you feel we have to get updated in recent time? I didn't understand the questions properly, so if you can explain me a little bit what you are looking for. Yes, so the technologies are changing every day. So is drug discovery in a recent time. And we are, as I said, AIs has been heavily got involved. Also, our analytical skill sets, tools has been improved in a tremendous level. So today, a great example is a nitrosamine, which is a very we can do in a LC-MS in a ppm level, which is very, very sensitive. And for me, the AI tool is AI will definitely change our world in terms of the timing, how much time it will take to compress our drug discovery. In fact, you are probably seeing some of those things happening in the vaccine development today. So hopefully I was able to answer your question. Any other questions?
Any questions from students? Don't hesitate.
So if any students have any question about the career, pharma career, or how you feel like market, how the pharmaceutical market or anything, feel free to ask about that also. It doesn't have to be related with the presentation.
Any questions? Sir. Good evening, sir.
Yeah.
So recently we have attended the webinar. In that, the professor told that based on the research, they found there are variations in the biosimilar products. They have compared three
Yes.
products, innovator product and three other biosimilar products. Among that, two of the biosimilar product are having different bio activity. So it is possible, or
so I don't know what studies they have done and what product they have taken. Different market has a different regulatory body has a different criteria of approving biosimilar product. So if you ask me, if the product has been taken from US FDA approved biosimilar, and then my first take would be, if he's saying they're different, the question becomes that I would question that whether it is actually fundamentally affecting the patient in terms of the efficacy, or is it just a minor differences on a protein that inherent to a biosimilars. So I would, without knowing the study, I cannot comment or challenge the study, but it depends on the market. But if it is coming from, let's say, kind of non-regulated market where the definition of the work is not very defined, then yes, it could be possible they are extremely different, and we have to be careful on that. But from a regulated market perspective, it is the question is whether it has any impact on patient or not. I would not challenge. I would not be that sure if it is an impact. At the end of the day, everybody, we are calling it biosimilar. We are not calling it bioidentical. So that definition, the term itself has its own self-explanatory.
yes, sir.
Sir, good evening, sir.
Yes.
This is Rajabandi from AKCP. Sir, while the formulation study from pilot scale to large scale, the formulation study is same, sir, or you have to change the formulation study?
Can you repeat the question? I'm trying to understand the question.
While we switch over from pilot scale preparation to large scale preparation, the same formulation study we follow, or that will be changed, sir?
So it really depends on the product, right? So if I am going to a pilot scale and my team anything develop, we may have done little bit of modification, but mostly we focused on the scale process optimization at that time. So we use different tools and to kind of say, okay, we are gone to this formula. The formula looks good. This is a weight granulation. We are going to use fluid bed granulators, and we are going to use a compression. We are going to use a B tooling. That premises in the formulation pilot scale will remain same. What we may have done, we might optimize the drying temperature. We might optimize the little bit more granulation fluid thing because it's so the fluid amount might have been different, So that's the only change I would usually do. I don't change the formulation in scale up because that's too many variable at that point to make the product working. Did I answer your question?
Thank you, sir. Yes, sir.
Okay. So online, someone asked me, what is the recent wide opportunity generic market, which market, so that we can prepare ourselves. Please let us know which job opportunity in more and demanding. So that's a great question. Look, India is a powerhouse of formulation development, and every market, India not only give supply to regulated market like US and Europe, and they also give product to the India and other countries. So I think that it doesn't matter which field you go. You might want it to, you can be a formulators, you could be a great analytical person, you can be a great packaging person, or you can be a great manufacturing person or a QC person. So one thing you need, anybody who is going to the industry, before going there, they have to, one thing have to be very careful. They have to understand what they're good at it. Are they good at chemistry? Are they hands-on on the equipment? Do they like doing, using, playing with the machine? Or are they like the chemistry synthesis? Are they like the all the reactions and all this thing? So that will drive the person, because at the end of the day, it is what you like eventually will tie up with what you will be good at in the future. So any job, there is a scope for every job. So I wanted to be very specific on that, I will not say you will only, if you do become a formulators, you will have the greater career, and you will not have a, if you are analytical, it's not a great career. Everything has a great career. Demand. So what we have is the market is a demand for the good technical person who is good at its skill, and it really depends on what someone likes it. So I hope that's answer your question. There's a question on the, sorry, Somia asked me a question. Which course may be good choice for student to pursue after PharmD career settlement? Okay. So can I am not very familiar with the pharmacy D program. Is it a doctorate of pharmacy or diploma in pharmacy? Can someone clarify that?
It's oriented towards clinical pharmacy.
Right. Okay. Thank you. That's what I thought. So if you are taking PharmD programs, you are already kind of decided that you are going to try to clinical side of the work. On a clinical side, one of the things that I personally think that has a huge potential in India is going into the pharmacovigilance side of the work. So the pharmacovigilance is a very upcoming system into the India. A lot of companies are outsourcing the work in the pharmacovigilance. In India, there's a company startup happening every day because of the scope of this thing. There are a lot of big companies are moving, setting up like a TCS, Accenture, and even they are setting up their center. So on a clinical side, that could be a good scope. Again, I kind of stay far away from. I'm just seen what I have seen into the industry, so that might be one place to go there.
Thank you, sir. So there is one more question.
Yes, please.
So nowadays, if you see the student, they will complete their B Pharm and they will join any of the job, marketing or industry. And most of the students, they don't want to study. They want to go for higher studies. They are settling with B Pharm itself. What's your thought on that, and how the higher studies can impact their careers?
So I don't know any other way to tell this story, but tell my story. So hopefully that will give somebody some message. So when I joined my B Pharm, I hated my B Pharm. I will be very honest with you, all of you. because my teachers really didn't give me any inspiration that I wanted to be into the B Farm. So I decided to do a marketing job. So I took a marketing job for fairly, it was a very attractive marketing job. And after joining a month, one thing I learned that this is not something I wanted to do forever. So this is something I would say every student has to understand. If you have to get an idea that very quickly what you wanted to do in your life very fast. So I knew that that was not something I would like to do. And then I said, you know what, I'll give another try on the science. So I came back. I went to the school. I did my master's degree, and I found my call. And at that point, even at that point, I realized that I need to learn really more. And so I had an option to go to the industry at that moment, but I decided to move on, and I kind of applied for the scholarships and came to US to do my studies. So I actually, and basically I will tell you what I did. I used my marketing times salaries to pay for my ticket, and I got the scholarship to come to US. My family didn't have money to pay for without scholarship. So that's my story, and I think that it paid me handsomely doing the higher studies. I wouldn't be here if I wouldn't have done the higher studies. So for me, higher studies is the only way in this field to achieve where you are. But you have to have your call for it, you know. And the only thing I can say, sometimes you might find one teacher for the students. You might have one teacher among all the hundreds of teachers you will find in the school who might give you the inspiration. So try to find out that one teacher that will give you the right direction for the higher studies.
Thank you, sir. Thank you, sir.
Yep.
Any other questions? So I think can you tell about the scholarship, sir? So can you tell about the possible scholarships for higher studies? So how they can proceed, or where they can search?
So when I was, and I'm long time ago, and usually it depends on your GRE score and your application merits and how much you scored in undergraduate, how well you have done your GRE score. Every college, master's programs, a good college has the student scholarships programs as a graduate student. And what I would say that there are certain expenses associated with that application. So you have to pay for your GRE's fee, your TOEFL fee, and your application fee. So you target 4-5 target and do a good research, right? So every college, whether you go to University of Iowa, whether you go to Washington University, University of Southern California, you can go to Tennessee, University of Tennessee. They all provide, depending upon how good is your score, how comfortable you are with your GRE score. So the student has to be very practical. They have to have they cannot only go to the highest colleges. They also have to go to the lower ranking colleges to get the scholarships, because sometimes scholarships can be achieved in lower ranking colleges. And sometimes people give partial scholarships. So one of the things a lot of colleges does, they say, we'll give you scholarship, but we'll give you 50%. You come to US and then we'll manage it. So sometimes people should take that chances, because what happened is once you go to the colleges, if you're really good, professor usually managed able to give you the remaining scholarships. And I only know about the US. I really don't know about the other parts of the world. I know the Canada is a little bit difficult to cover the scholarships because there's a in-state tuition fee versus outer-state tuition fee. So Canada is a little bit difficult. And I really don't know any other country. But I think in US, if you do the GRE well, TOEFL well, and your undergraduate scores are good, and one of the things you definitely people should do, try to get some publication at the undergraduate or a master's level itself. The publication helps. A good publication in an international journals definitely helps. I had one when I was applying from my master's thesis, so that definitely helped me. And also make sure that you have a very clear, precise idea what you wanted to do with your research. Don't jump into the different field and use the same story. So those are the few things. If someone does that, that's a secret stories. Everybody can get the scholarship. I don't see any issues. I told the same thing to many students many years, and it worked so far.
Thank you, sir.
Thank you. Thank you. Thank you.
Okay, so I see there's a one question came in that someone asking that how to get a formulation job in US while you are working in India. So yes, so there are two sets of problem right now. The US H-1B visas rules has been really become very difficult. So that itself has its own problem. In my past, I have taken people straight out of India to join the US on H-1B. That segment that window getting shorter and shorter and shorter, and it's a really difficult market. So one of the best options is that you join an arm, which is a MNCs, which has a presence in US, and try to get into L1 through them, and that L1 visa, that might be the only way recent time you can get it to coming to US, at least till the political system in US changes and economy comes to a better position in 2-3 years from now. I think that in 2-3 years there will be a lot of more opening in the pharmaceutical operations in US, and people who has good operation backgrounds will be benefited out of it. Okay. Any other question? I see someone just wrote something. Is if the scholarship available for any postgraduate course? Can you mention any course name? Any course is available in India, and Indian government can avail any scholarship postgraduate in medicine field. Okay, so scholarship postgraduate course. So I'm assuming that you are asking about the US scholarships. The thing I just told before, it is primarily focused on US scholarships, and these are only focused on master's and PhD degrees. So the scholarships available in the United States is primarily focused on PhD programs, and these programs could be a fairly long-term programs. It stands between 3 to 5 years commitment that you have to give, and it is what it is, and that's what you have to know that if you take it from the US postgraduate program. So definitely the most scholarships are all related with the PhD programs, and they are really available. So as long as your undergraduate score and the GRE score and the TOEFL scores are good. Any scores available in Indian government for any, I'm not sure I understand the questions. I'm assuming that if I am aware of any scholarship program or not, and honestly speaking, I don't know anything about the government of India's scholarship program. So sorry about not to be able to answer that question. Okay, so I think we are almost towards the end, right, Dr. Venkateshan?
Yes, sir. You can please continue.
Okay, so I guess that's a questionnaire, right? So, so my side, thank you very much for inviting me. I really enjoy the questionnaire session. That's where I really have the fun. So thank you for all the questions, and thank you for inviting me again. It was being real fun.
Sir, sir, in this occasion, we are expecting your cooperation and support in future endeavors, sir. Whenever you would like to plan to India, sir, particularly in Tamil Nadu, please let me know, sir. We will invite you in chief guest, sir, in future also.
So how far you are from Chennai?
Overnight, sir. Overnight, 8 hours.
Which part of to Kerala side, closer to Karnataka side? So tell me a little bit.
Madurai, sir. From near Madurai, sir.
Oh, okay. So that's
40 minutes away from airport, sir. 45 minutes away from airport, our university.
Okay. I will try to come. I do come to Chennai.
We will arrange made all the things.
Okay. I will definitely try to come. I have a plant in Mysore area, so if I am visiting there, I will definitely try to come. But till the whole COVID-19 thing has been resolved, I don't know when I'm traveling back to India. So
next
it
year,
might be
whenever you plan. Next year, whenever you plan. Okay.
Sure, absolutely, definitely.
In future also, let's have a good collaboration, sir, for the betterment of our students' career.
Absolutely. Anything to help the student.
Yes, sir. Take care, sir.
Okay. Thank you very much, everyone. Have a good evening.
Thank you, sir. Bye, sir. Bye.